Hello, everyone. I'm Wendy Shelton from Intellos, and I've been delegated to welcome you to our webinar and introduce today's presenter, Dr. Caitlin Beaver. Next. A little bit of information about our webinar technically. Your phones will be muted, so if you speak to us, we will not hear you. If you could be careful to not have your audio on your computers, that would be very helpful to prevent feedback. Intellos will post copies of this presentation, including an audio and the slides, on our website, or at least there will be a link on the website so that you can hear this again and pass it on. We're going to be taking questions at the end of the presentation, and use the chat function, the typing chat function on your screen to the left, and we'll do our utmost at the end of the presentation to answer those questions. Today's presenter is Dr. Caitlin Beaver. She's a dynamics engineer at Intellos, and most notoriously has just presented a poster at the American Heart Association Scientific Sessions just a week ago, and that is the basis of our presentation today. Just to give you a little bit more background, for those of you who don't already know, a little bit more, next slide, about Intellos, and that is that we do work in many different areas of physiology and are leaders in physiologic mechanistic modeling and simulation. I think today's presentation you will find is a really lovely example of how simulation can work side by side with in vitro and in vivo analyses to really enhance our understanding of physiology. We work in multidisciplinary teams of engineers and biologists in order to come up with these complex models, and we've been doing it for quite a while. These are many different disciplines we work in: dermatology, metabolism, and today we're going to be talking about cardiovascular studies. So without further ado, I'd like to pass the microphone over to Caitlin, and look forward to your questions at the end.
Good morning, everyone, or good afternoon, depending on what time zone you're in. Just the original genesis of this talk was to describe the results that we presented last week in a poster to the American Heart Association annual meeting, as Wendy mentioned. And specifically, that poster presented some simulation results that we had derived with one of our mathematical models that we call Physiolabs. But as I was putting together this presentation, I realized that the results themselves were only part of the story, and that it was actually equally interesting to see how our learnings grew out of the way that we build models and how the story demonstrates some of the advantages that modeling brings to the table over pure experimental research. I structured this presentation into 3 sections. So we'll begin by going over some of the background principles in the development of Physiolab models that are common to all the work that's done here at Intellos. I'll then provide you with an overview of the human cardiovascular Physiolab, which has been one of our real workhorse models over the years, but that we continue to evolve to capture the latest scientific information on cardiovascular disease. And just a note that throughout the presentation, I may alternately say CV when I mean cardiovascular. So the CV Physiolab provided the basis for the development of a model of plaque growth in a genetically altered mouse, also known as the ApoE knockout mouse Physiolab. And I'll finish up the talk by describing the whys and the hows of that transition from human to mouse model and some of the things we learned about the biology along the way. At the end of the webinar, as Wendy mentioned again, we'll open up to a question and answer session with the audience. So with that, let's begin. So Intellos as a company specializes in predictive modeling of disease. We provide products and services that enable robust decision-making for the prevention, detection, diagnosis, and also treatment of these diseases. And we do this through the development of large-scale mathematical computer models of biology. A reasonable place to start today's discussion is to go through how we build these models in order to answer the questions that our clients bring to us. So our general approach is referred to as the top-down method of model development. And what that means is that we start by considering the most macroscopic aspect of any disease before working our way into the details. If we take the cardiovascular Physiolab as an example, that top-level description consists of clinical outcomes. So we begin by identifying the key clinical measures related to cardiovascular health, such as LDL cholesterol or intima media thickness, and we define the data that our model will be required to reproduce with its simulations. The next level of research is to determine the key aspects of the biology that relate to clinical outcomes. So again, referring to the cardiovascular physiolab, if LDL cholesterol is a clinical outcome, then we need to understand how particle catabolism leads to a particular particle concentration. So what are the dynamic interactions that lead to the clinical output? Next, we determine the key mechanisms that regulate the important rates at the level of detail necessary to reproduce those clinical measures and biomarkers. And finally, once we have addressed all of these levels of the biology, then we can assemble the information to create alternate virtual patients that cover known clinical variability. And when I use the term virtual patient, I mean a parameterization of the model that represents a single unique individual. So the structure, also called the topology of a model, is the same for all virtual patients, but the parameters for each one vary, just as genetic differences in real humans lead to differences in our physiology. In the history of Entellus' work in predictive modeling, we have addressed a number of different problems with a variety of partners in industry, in government, and nonprofit organizations. And the modeling that we do is able to help us answer questions at all levels of the drug development pipeline, from target evaluation all the way up to clinical trial optimization. So I'm not going to go through all of these different examples on the slide, but I would like to highlight the application to translational medicine that is of particular relevance to the work presented today. So we can use models to gain an understanding of the impact of interspecies differences in disease biology on the differences that we ultimately care about, which are why do some therapies show a response in the preclinical setting and then fail in humans, or alternately, are there therapies that would be useful in humans but that we're missing because we don't see an effect in animals? So with that background in mind, let's dive a little deeper into the human cardiovascular physiolab. The human CV physiolab actually has quite a long history at Entellus. The platform was initiated in 2005 when we built a model to describe the dynamics of cholesterol metabolism with the objective of evaluating the efficacy of C-step inhibitors for altering lipid profiles. We continued to add to the model, and by 2007, we had an extended platform that includes the description of atherosclerosis, or plaque development, in the human coronary artery. At the same time, we worked with some academic collaborators to build a finite element model of plaque stability that allowed interpretation of plaque status measures in the model in the context of the risk associated with plaque rupture. And since that time, the model has undergone a number of further updates, such as the addition of platelet and endothelial cell biology. We've represented both erosive and rupturous plaques, and we've also expanded on the types of virtual patients that are represented. And additionally, we developed a mathematical framework to link virtual populations, so that's essentially a collection of virtual patients, to well-known risk scores such as Framingham, that make statements about the relative likelihood of having an adverse cardiovascular event, such as a heart attack. So work on the cardiovascular physiolab is ongoing, and our current efforts are focused on detailed representations of type 2 diabetes, as well as circulating inflammatory biomarkers. So here we summarize the main features of the CV Physiolab. So in a nutshell, the CV Physiolab is a mathematical model that integrates information about all the important biology that leads to changes in cholesterol profiles, changes in the rate of arterial plaque growth, and ultimately determines what these changes mean for someone's likelihood of having a heart attack.